Okay, we're gonna start with the ninth talk from this Solanaceae seminar. Hello, everyone. As every Friday, we had today a very special speaker and a very interesting topic. The title of the talk today is Implication of Phylogenomic Analysis for Macroevolutionary Patterns in the Large Genus Solanum, Solanaceae. And I'm very excited to present today to Edeline Gagnon, our speaker. Today, Edeline completed a master and a PhD degree between 2009 and 2015 at the University of Montreal in Quebec, Canada, under the supervision of Dr. Anne Bruno from the University of Montreal, and also working with the Dr. Colin Hughes from the University of Zurich in Switzerland, with a focus on systematics of Fabaceae, but with a strong interest in taxonomy and biogeography. Her PhD was focusing in the systematical macroevolutionary patterns of the Caesalpinia group, that is a group of 225 species of pantropical woody legumes, where she carried out a taxonomy revision of the group, describing 5 new genera and 75 new nomenclatural combinations in the group. That was amazing work. After a 1 year non-tenure contract working as professor at the University of Moncton in New Brunswick in Canada, between 2017 and 2018, Edeline moved to Edinburgh to start her postdoc at the Royal Botanic Garden, Edinburgh in Scotland, working with Dr. Tina Sarkinen. And Edeline will give us a talk today, and she's gonna show you the work that she's been doing for the last 2 years, and where she was very interested in investigating the patterns of niche evolution in Solanum. And Edeline, thank you for accepting this invitation. Can you share your screen, please? Thank you, everyone.
Thanks, Andres. So I'm just going to take a moment here and share the screen. Right, so everyone can see that, and everyone can hear me.
Yeah.
So I guess I'll start. Okay. Thank you for inviting me to present in this seminar. It's a real honor to present. So, as Andres mentioned, today I'm going to be talking about some phylogenomic and macroevolutionary analyses that I've been conducting in the genus Solanum. And as Andres mentioned, I did not start out my career in Solanum, but to explain a bit why I decided to cross the pond over in the UK, I just want to go back a bit with what I did actually in my PhD at the University of Montreal, where I had this amazing opportunity to pursue a research project looking at the systematics and macroevolutionary patterns in the Caesalpinia group in the Leguminosae. So this is work that I was super proud of. I sort of investigated patterns of biome conservatism across this group of 225 species, looking at also switches in evolution of habits, which seem to be correlated to shifts in evolutions of biomes. And while this was super interesting, as it often happens at the end of a PhD, I was actually frustrated by some of the aspects of my research, namely by the fact that we were using biomes, because as some of you might know, biomes are notoriously complex concepts. So just as taxonomists, we have these multiple species concepts that are all elaborated around the fact that we can't really agree on what are the best criteria to delimit species, because it can depend on different types of organisms or different types of evolutionary patterns ecologists often struggle with the same sort of problems in biomes, in that you can, the selection of how to define biomes can often be related to different geographic scales, and as such, I actually think that understanding what are the barriers that lead to these creations of specific entities or biome entities might actually be the more interesting question. And so I really wanted to keep exploring questions related to macroevolutionary analyses, but using a quantitative approach rather than a qualitative approach. And this is what led me to be interested in in Solanum. So Solanum is a super interesting genera because, well, it's one of the top 10 biggest genera in angiosperms. So it's about roughly half the diversity in Solanaceae, with 1,248 accepted species. But there's also this amazing database in Solanum that's available through the work of numerous taxonomists, which have been contributing to Solanaceae source over the years, resulting in the fact that we have this georeferenced occurrence record database of more than 91,000 vouchered herbarium specimens. So this seemed to me like an amazing research source in which to ask some quantitative macroevolutionary questions. Solanum is also super interesting from an evolutionary perspective because it's morphologically diverse. We find them as shrubs, herbs, lianescent vines, trees, and even epiphytes. And they're obviously important, of course, because we also have lots of crop species in Solanum, the most well-known which are potato, eggplant, and tomato. But I also recently found or compiled a list of 24 non-model crop species that exist in this group. So in terms of macroevolutionary analyses, I became really interested in the evolution of geophytes in Solanum. And the reasons are twofold. First of all, when we think about the evolution of habits in plants, we often neglect the underground biodiversity of plants. We tend to focus on the upper parts rather than the below ground parts. And Solanum is obviously famous for potatoes, so Solanum tuberosum down here. But there's actually a whole range of different geophytes in Solanum. So you actually find species that have these tuber-like appendages or that are also rhizomatous. And so the repeated evolution of these growth forms is an opportunity to test ideas about convergent evolution and in independently involving lineages. And so we do find these different forms in Solanum, but it's still not very well understood in the literature whether, for example, rhizomes and tubers, do they occupy the same ecological niches? What are the evolutionary pressures that lead to the evolution of these different growth forms? And so I've been designing a study where we're trying to answer these series of questions. So in Solanum, are species with tubers and rhizomes different in ecological and geographic space? Is the evolution of tubers or rhizomes a dead end to evolution, or what exactly is their evolutionary lability? And finally, do tubers and rhizomes promote niche shifts, allowing the genera to evolve into new niche spaces or geographic areas? So the way we've been approaching this is that I've taken that database of occurrence records in Solanaceae source, and I've been cleaning it with custom R scripts, so written in-house. We found lots of mistakes. We corrected them and have been excluding data and also doing some spatial filtering analyses to avoid sampling bias in our data sets. So this led me to compile an occurrence record with 53,026 records. So that had about 90% of the accepted species in Solanum, which is pretty good, pretty interesting. And then I asked the help of my colleagues, who are the real Solanum experts, to help me code each species to see if they could be considered as non-geophyte, tuberous, or rhizomatous species. And what you see here is a map where in red we have all the occurrence records, and we actually have maps of where tuberous species occur in Solanum across the world, and where rhizomatous species in Solanum occur across the world. Whoops, sorry. And so we found some super interesting geographic patterns emerging. So the tuberous species being predominantly found around the Amazon and up Central America into the middle of North America. And this unexpected pattern here, down here, where rhizomatous species in the neotropics seem to have amphitropical distribution, and this huge predominance of rhizomatous species in Australia. So that's sort of answering the question that tubers and rhizomes don't really occur in the same geographic space. So the next step is we can actually characterize the abiotic niche of these different growth forms by taking the occurrence data points and extracting environmental variables from maps. So we can find online these bioclim map variables related to different temperature and precipitation variables. We decided to go and select the ones that were ecophysiologically meaningful, meaning that they are related, they have an impact on the growth and survival of plants. So focusing on variables such as that are related, for example, to temperature, frost, drought, temperature, precipitation, seasonality, growing season length, and even variables related to fire, which cause obviously disturbance in plant growth. So now that for each occurrence point we had these different an environmental value, we can start sort of exploring whether these different growth forms have different ecological niches. And so our expectations, for example, are that rhizomes and tuberous plants, because they have these extensive below ground parts, it's sort of a response to, it's an evolutionary response to disturbance. So we would expect, for example, to see them occurring in areas where temperature, harshness, and seasonality is stronger, where there's more drought, where the growing season length is shorter, and where there's more fire. In the first step to sort of explore our data, I first did an ordination in PCA environmental space. So as you can see, 53,000 occurrence points in the PCA space is very, very hard to interpret. Basically, it looks like a giant pizza. But to be able to understand actually the distribution of these points more clearly, we apply these kernel density estimation methods. And basically what you see in these graphs is similar to here, except that we sort of have a heat map. So the darker parts are the areas where you see more strongly a denser occurrence of points than the paler parts around here. And we can even draw contour lines corresponding, for example, to the 50th, 90th, and 95th percentile of the density of the data. And so here what we can see is that in yellow and red, we have the occurrence of all the Solanum data we have. In blue here, we have the density occurrence in PCA space of the tuberous data. In black here, the non-geophytes, and in red here, the rhizomatous species. And as you can see from these figures, the tuberous species are not occurring as densely in the same spaces as the rhizomatous species or the non-geophytic one. So it's some indication that they are occupying different ecological niche spaces. So you can actually go further than that. And I'm sorry, these are all still preliminary figures because I haven't had the chance to complete these analysis yet. But it's my intention to actually calculate niche overlap amongst these different growth forms in environmental space. And so it's possible to do so notably through this framework developed by Brunneman et al. In 2012, where they use this, so they use this sort of same, these same kernel density estimators and transform the environmental space into PCA. It's possible to do this through the package EcoStat. In addition to actually calculating overlap in this PCA space amongst the different growth forms, you can also look at the density of occurrence of these different growth forms along individual niche axes using that same package. And so here I've put some interesting preliminary results. Notably, we have here the environmental niche axis representing frost, and the environmental niche axis representing aridity. And so the line here represents the density occurrence of all our Solanum data. And in the green curve here shows actually the density occurrence of tubers, and in red, we have the density occurrence of rhizomes. So you can see here that that while the majority of Solanum seems to occur in this frost-free environment, the density occurrence of tubers is actually more strongly, there's actually a higher density occurrence towards temperatures that are below 0. Which is not the case, though, for the rhizomatous taxa. And similarly for aridity, the tuberous species seem to be in areas that are more moist than, for example, the rhizomes, which are really occurring in dry environments. So again, it's showing us that the tubers and the rhizomes are possibly responses to different environmental stressors. One of the neat things you can also do with this type of analysis is rather than comparing growth forms, you can also compare geographic areas. So I've got similar graphs here again, but here this time the green line is actually all the Solanum data in Australia, and the red line is all the Solanum data in the neotropics. And I actually wanted to see, because we saw that in the map, the rhizomes were occurring in Australia as well as in the neotropics. And I wanted to see if they were responding to similar environmental pressures. And we actually found this very interesting pattern where the non-geophytes in Australia and the neotropics seem to be occurring in environments where there was less or smaller fires, whereas the rhizomatous taxa seem to be occurring more frequently in areas with bigger fires. And that to me is a really cool result because Solanum taxa in Australia are actually known as fireweeds. And so we're sort of, I think this is sort of a demonstration that rhizomatous species are actually potentially adapted to disturbances by fire. So I'm still working on these analyses, but to answer the questions 2 and 3 from these from this study, so how many times did rhizomes and tubers evolved, and I'm interested in also applying these tests for niches in the abiotic environment and whether these are associated with traits. And these require phylogenetic comparative analyses. So we do have fine scale, high quality taxonomic data and occurrence data as well, but we still need a better evolutionary framework. And this is because the most recent phylogeny of the Solanum group, which was published in 2013 by Sarkinen et al. In BMC Evolutionary Biology, only contained 34% of all Solanum species. And so while I have carried out some preliminary, for example, ancestral character state reconstructions with this phylogeny, there's always the doubt that because it's so, the sampling is quite low, below 50%, we're not sure about how confident we can be in the results and taking conclusions from these results. So we decided to build a better phylogeny for Solanum, which is not easy at all because Solanum with 1,248 species is quite large. For those of you who don't know Solanum, the next slide here is sort of an overview of the major clades. And so basically, a simple way of presenting Solanum is you first have this early, first diverging clade called the Theloponium clade with 3 species. And then the entire genus can be divided in 2 clades. So clade 1 in blue and purple here, which contains mainly herbaceous non-spiny Solanums, which include the potato clade and the M clade. And then clade 2 here in orange, which contains for the majority these shrubby spiny Solanums. So to move forward on this and construct a supermatrix phylogeny, we need to collaborate. And so in Tina Sarkinen's lab, I was fortunate enough to be invited to participate with the MSc student Rebecca Hilgenhoff in the construction and finalization of a supermatrix phylogeny, which took all available sequences published from GenBank, as well as sequences from collaborators in the United States, South America, and Europe. Sorry, something's going on with the PowerPoint. And putting this data together, which used 2 nuclear and 7 plastid loci, we actually built a supermatrix phylogeny which contains 60% of all species in Solanum, so including 325 previously unsampled Solanum species. So to make a short story, the results from this phylogeny were pretty congruent with previous results. So this next slide here shows if the name is highlighted in red, if a name is highlighted in red, basically either it's a minor clade that received poor bootstrap support or whose circumscription changed. And as you can see, it doesn't occur very often. So that's a good sign, meaning that inclusion of more data doesn't lead to profound changes in the taxonomy of Solanum. Great. The data does show some newly identified clades, which are mostly specific, and it includes Solanum anomalostemon, Solanum polygamum, and Solanum euacanthum. There's also this clade that's the trachytrichium clade that's not really new. It's actually is a clade that's discovered by Dr. Leandro Giacomin, who's a specialist in section Brevantharum, and who kindly agreed to lend us his data. So he will probably talk about it in the next, in the forthcoming Solanaceae seminar on the 30th of October 2020. So this is all I'm going to say about the taxonomy of Solanum for now, because in 2 weeks' time, you will have the pleasure of hearing a talk by Rebecca Hilgenhoff, so the MSc student with whom I worked on this phylogeny. And so her talk will focus more on trait evolution within Solanum, so looking at whether we can identify snapomorphies for all of these clades. And she'll also be talking about a really cool online multi-axis identification key that she's been building for Solanum. So that supermatrix phylogeny is a huge achievement. It's like, and you can say, okay, Edeline, so now you can go back to your macroevolutionary analysis. But actually, I wanted to push the phylogeny a bit further, because as we all know, there's been some recent advances in next generation sequencing that have led to some profound changes into how we build and view phylogenies. Notably, there's the way we've been going around with this supermatrix phylogeny. Well, we know it's a bit old-fashioned because we're only looking at a few trees at a time, and we know that the species trees of a group is really composed of a forest of gene trees, which I learned is called a phylome. And with next generation sequencing, what it allows us to do metaphorically is to visit a forest of the species trees. So to look at all these gene trees at the same time. And this is great because it gives us more data, but it's also a big challenge because these different gene trees can give us different stories, and it can be difficult to evaluate what's the correct phylogeny. Furthermore, the other big challenge is that, as we know, we tend to represent evolution in this bifurcating matter, but the more recent studies have shown that species that discordance in gene trees is often prevalent and might be an indication of reticulate evolution. And that has huge implications for taxonomy and comparative biology. So in order to investigate these questions, what I did is I decided to compare the supermatrix phylogeny we generated with a plastid and a target capture phylogeny. So the plastid phylogeny we generated using genome skimming. And so we added 83 samples and supplemented this with data from GenBank. And for the target capture phylogeny, we had a smaller sampling of 13 species. I'd like to thank Angela McDonald and Christine Martin for collaborating and sending us about 15 to 18 samples, which we added. And then we took another additional 12 samples from GenBank. The bait sets we used were the 353 angiosperm bait set from the Paftal group and Q. So now, obviously, the plastid and the target capture data have less samples, but we still wanted to see how their topologies would compare with this more densely sampled supermatrix phylogeny. So as we know, the influences of analysis parameters can have a huge effect on the recovery of species tree, including, for example, missing data in taxa, data partition strategies, and the different types of phylogenetic analyses as well. So I designed this study where I modified or varied all these parameters and compared the topology at the end of 36 different species trees. So the results that I got from it is, first of all, the first result is that most major and minor clades are recovered in the plastome and target capture phylogeny. I can't say much more because obviously these plastome and target capture phylogenies have a much smaller fraction of sampling than what's included in the supermatrix. But it's still a very good and positive result because it's suggesting that the monophily of certain intrageneric groups are becoming truly robust across all three of these data sets we compared. Key result number 2 is that we found that the type of phylogenetic analysis we did had a huge influence on the recovered species tree. So you can sort of see it here. This is a PCA of Kendall-Collinge metric, which is it's based on, so we take the topologies of the different species trees we find and then build a distance matrix. And so it sort of shows how different the species trees are from each other. And you can see here in green and gray, all the coalescent trees tend to cluster together. And up here in black, yellow, and blue, the maximum likelihood tree clustered together. Other surprising result as well is that the backbone of the supermatrix tree was not recovered in any of the species trees that we got from the plastid and target capture phylogeny. And you can see that here with the supermatrix tree up here in red and differentiated from everything else. So to show you what exactly were the topological differences, I have here, so I pruned the supermatrix tree so that it had the same number of tips as the plastome tree. And basically in the supermatrix tree, we found clade 1 here with the potato and M clade sister to clade 2. If we look, however, at the plastome tree, we find differences. So we recover a monophyletic clade 2, but then clade 1 here is divided. So up here you have the potato clade, and down here Regmandra is no longer a sister to the rest of the potato clade, but rather sister to the M clade, and together they're a sister to clade 2. So this is quite a big difference, and you can see down here that, for example, the sister relationship in this tree between Regmandra and the M clade is quite strong, but sister relationship to clade 2 is very poor. But actually, when you look across all the different species trees, these numbers keep changing, and this backbone actually keeps changing a lot. You might be wondering what happens with the target capture data. Well, we sort of get a similar story, but it sort of gets worse in the sense that the M clade is not recovered here at as monophyletic. It actually splits furthermore into 2. So this leads me to ask, the question about which of these phylogenies actually represents the true evolutionary tree. I wanted also to show you a bit the backbone for clade 2 because you have similar problems where, for example, here we have in the supermatrix phylogeny Anomalistemon, which is sister to the rest of clade 2. But in the plastome phylogeny, we recover with strong support as sister to the rest of the Brevantharum clade. And similarly, you have other groups that keep floating around, for example, Scyphomandra, which is not a same position in these two trees, as well as the minor clades here, Caroliniensis and Hieronymi. So in the plastome data, they're found as sister to Elegnifolium and the old world clade in the Leptostemonium. And I've just put here the target capture tree, but as you can see, the sampling here is quite sparse, so we can't come to any strong conclusions again. So what is the degree of the gene tree conflicts that we've been observing. So I've been using a software called phyparts to visualize this gene tree concordance. Basically what it does is that it goes, I feed it the species trees and the gene trees, and then for each node, it calculates the number of gene trees which are in concordance with that node, in conflict for a single dominant alternative tree, or in conflict, so proposing a completely different topology, or in gray here without any information. And so as you can see, before I've even zoomed into this plastome phylogeny with these five parts nodes, it's predominantly red. So suggesting that there's a lot of gene tree discordance all across this plastome phylogeny. And here I've simplified this, this figure. So I've collapsed some of these clades here. And so luckily, Solanum here, there's out of the 77 exon genes that I examined for this phylogeny, a majority of them are suggesting that Solanum is monophyletic, which is great. But then for other key nodes, for example, here the relationship between Regmandra and the Morolides here, there's only one out of our 77 loci that we fed into this analysis that had a gene tree that was concordant with this. So whether this relationship is real or not, that puts a lot of doubt into it. You might be again wondering what's going on with the target nuclear data set. And so I carried out the same 5 parts analysis here. And there's a bit more blue in these nodes, but I have to admit that we were incredibly surprised when, if you look down here near the base of the tree, where we've been having lots of problems, that there are, so the numbers above indicate the number of concordant gene trees, and below the number of conflicting gene trees, and then here we have 0. So we're wondering what's going on here. Is it the analysis, or did I do something wrong? So I actually decided to use a different metric of gene tree concordance, which is a method that's implemented in IQ-tree that's quite similar, that calculates a gene tree concordance factor. So based on the gene trees using the analysis, but also you can also calculate a site concordance factor. So using the raw alignment to see how many sites in the alignment actually concord with the species topology. And so there's some things that are reassuring here, in that the nodes in 5 parts where we had a high number of concordant gene trees also have high percentages of gene tree concordance factor and site concordance factor with this method in IQ-tree, as you can see here in blue. But again, the main areas where we find the most discordance amongst the gene trees is up here in the eggplants, Leptostemonium clades, where often these concordance and site concordance factors fall so below 20%, I would say, for the GCF, and below 50% for the SCF. And the other area where it's quite low is here towards the base. So this is really interesting, because in a sense, up here the eggplant clade is somewhere where we would expect a lot of gene tree discordance, because a recent paper in New Phytologist that just came out showed that this is the area where there's the highest diversification rates in Solanum. So it's a really fast evolving clade where there is the potential to have a lot of incomplete lineage sorting. So we might be in the presence of soft polytomy here. So more data or more analysis might indicate that actually that it's possible to resolve a fully bifurcating phylogeny up here. However, down here, deeper in the phylogeny, where you have these, tend to have these very short branches, that's sort of generally much more hard to resolve. And I don't know, but my feeling is that we might be in the presence here of a hard polytomy. So we can do further tests. So for example, I have been carrying out these polytomy tests that you can do in Astral, that basically ask whether they have this sort of null hypothesis where the branch length is 0. And so a rejection of the null hypothesis means that the branch length is not 0, so we have a bifurcating branch. And I have carried them out on a plastome exon phylogeny and the target capture phylogeny. And an interesting, super interesting thing that has emerged is that we do, for example, in the plastome phylogeny, find a polytomy here at the base that involves clade 2, the M clade, the potato clade, and the spiny African solanums, but it doesn't involve Regmandra. And same thing here in the target capture phylogenies. There are indications of certain areas where the branch lengths might be equal to 0, so we might be presence of a polytomy, but again, it doesn't involve Regmandra. So so this may be one way forward if we want to get a better understanding of the phylogenetic relationships in the Solanum group, is to really target a transcriptome or some sort of genome analysis of this clade to get a better understanding of the early evolution of this group. And the other thing I'm currently pursuing is trying to build network phylogenies of this group. So I've so far only done splits tree, which is a distance-based method that allows you, so it allows you to sort of explore the data. And we can see that, for example, in the target capture data, there's a lot of reticulation at the base of the tree. Also here in the potato clade, among potato species, and amongst these species, closely related species here. So the next steps for me would be also to test if there's actually reticulation using PhyloNet, and also these incomplete lineage simulation sorting simulation tests. So we will see if more data and more analysis can really help solve these discordant or poorly supported nodes. If I go back to my initial question, which is what are the implications for comparative biologies? Well, I would have to say that hard polytomies are probably here to stay, and we are going to have to learn how to deal with these. And so I'm in my final year of my postdoc. I'm currently preparing to submit this phylogeny, this comparison of the supermatrix target capture and plastome phylogeny to the American Journal of Botany special issue. And then I'm going to focus on looking whether these traditional methods for accounting for phylogenetic uncertainty in phylogenetic comparative methods, are they really enough? Are they really, for example, taking into account all this variation I'm seeing across these different species trees? I'm also trying to apply for a Marie Curie fellowship. So I think that actually building a bait set directly for Solanum could be super useful to gain a better phylogenomic insight into the group. And we could also target loci subtending interesting traits and do all kinds of interesting analyses with this. So I want to conclude with my talk with this, because the deadline is for the 9th of September. I'm curious to see if anyone else is interested in this type of work. Yeah, so just give me a shout and let me know. And with that, I'd like to conclude my talk by acknowledging all the people who have collaborated with me on this project, all my co-authors, all the sources of funding. And yeah, please don't hesitate to, I'm ready to take your questions, and please don't hesitate to write to me. Okay, I finished.
Okay, thank you very much. That's great. That's super great. Oh, just Andres told me that he's having some problems with the internet. So I will be taking the questions. If you want to drop your questions on the chat or just to turn on your microphone and ask the questions, please feel free.
I'm just going to stop sharing the screen for a moment.
Diego Morales Briones just raised his hand. So if you want to talk, yes.
Oh, hey. Hi, Edeline. Yeah, it's a good talk. I really like it. So I might know what's happening in your HiveSeq dataset when you go to slide 40 and 39, where you have a lot of gene tree discordance towards the base. So the input gene trees for 5 parts needs to be rooted with halcomata. And when you have that pie chart there is because all your input trees was unrooted. So one way to verify this is that because every gene tree should be rooted with halcomata, actually the first pie chart needs to be all gray or all blue. If you use the filter for 5 parts, let's say that 50% bootstrap support, there's no label in there. So 5 parts takes that as 0. So all the pie chart needs to be gray in that case when you plot it. Or in the opposite, if you use no filter, every gene tree should be rooted with halcomata, and the pie chart needs to be all blue. If you don't have a root for your gene trees, you should not include it. So that's going to change completely your first 3 pie charts towards the base. Towards the tips is going to be, I'm pretty sure it's going to be the same, but that's the problem. Yeah. I just want to say that because I've used 5 parts a lot and I've seen this problem. It's because sometimes we forget to root the trees. So, and for...
I was...
Yeah. And
no, please, I'll let you finish and then I'll answer. Sorry.
And for IQ3, I never use, I use other things to do that, like quarter sampling. So make sure that for IQ3, you might need rooted or unrooted trees. That's another thing you need to check too. But
yeah. I have a question for you then. Is there a simple way to do that in like just changing a parameter, or do I actually have to go through it through my data manually to do that?
You need to root it every gene tree, whatever way you like to do it. But all gene trees need to be rooted, and the species tree needs to be rooted. And
so the species tree was rooted, but yeah, I didn't do it for the gene trees because I thought it would be, I had to identify each trees that had the root or not, and then yeah. So there's no easy solution to it. Okay, but that's super helpful. Yeah, thanks.
So and if you don't have the tree, don't forget you cannot use trees that you don't have the root. So that's why sometimes I added more roots, more outgroups, so you can have alternative ways to root the tree.
So you can simulate, like you could put a polytomy at the bottom.
No, I'm saying that, for example, for your data set, you only have 1 root, right? And if for some reason the hybridization didn't work, then you cannot root that tree. So then you cannot use it for 5 parts. So maybe you want to add another 2 or 3 outgroups or whatever. But yeah, that's my advice.
But I could still root it in the right direction, right? So I could still root it using the most, what I know is the most basal lineage.
Well, no, because then you fall into something that you think is the root, and then you might influence by chart. So is either you root it with halcomata or you don't use the tree.
Okay.
Or just add more samples for our groups. Yeah.
Yeah. Cool. That's super helpful. Thanks.
Thank you.
That's great. Thank you. I think Andres is back. So
yeah, I had some problems with my internet connection, but I'm back. If someone have more questions, please just turn on your microphone if you have some question. Your camera you want. Someone else?
I wanted to say that is it correct, Edeline, that if in the Marie Curie proposal, if somebody is already using bait sets, or that the bait sets would enable if designed correctly, to collaborate on exploring some particular research questions that would really enhance the Marie Curie, the European postdoc funding?
Yes, so thank you, Tina, for yes, bring that back up again. So I went really fast at the end because I know I was running out of time. But yeah, so the Marie Curie bait set, the idea is to not only target loci that would be interesting for phylogenomic analyses, but that would also be interesting to look at evolution of genes and loci that subtend interesting traits across the entire Solanum family. So examples of these could include, for example, disease resistance, evolution of to drought, to different abiotic stressors, trichomes, glycoalkaloid, glycoalkaloid chemicals, self-incompatibility genes, etc. So I'm currently writing up this proposal with the idea of really producing a tool that would be useful for the entire community. And taking advantage of the fact also that Solanum is one of these rare genera where we have 3 really good genomes with eggplant, potato, and tomato, and that there's been a high level of synteny that's been demonstrated across all 3 genomes. So there's really a huge potential there to explore these different questions, looking at, for example, genotypic and environment interactions and evolution across a mega diverse clay. So if this is something that you have thought about or are interested in, I would be really, I would love to hear from you. And I'm trying to—
So as an example,
yeah.
Am I right saying that even though you'd already designed baits and you use them regularly, if by including a smallish set of genes from your existing baits in your set, you then enable kind of cross-sampling for another project, you would in essence produce these for free, and the proposal is likelier to get funded because it's seen positively within EU.
Yeah, exactly. Yeah.
So yeah, if you have something that you're already researching, and I know in the audience there's phenomenal projects on various aspects, and if they are somewhat humble in their size, obviously not taking the entire bait system, do email or drop in. That would be really beneficial for the entire community, I suppose, because your sampling is very wide across the Solanium, so species that perhaps other researchers might not have access to.
Yep.
Okay, thank you, Tina. More questions?
Yeah.
No,
hi.
Zach, go ahead.
Okay.
Oh, thanks so much.
Okay.
Hi there. Really great talk. I
Okay.
wanted to ask a question that comes from maybe a naive perspective, because we work on Solanaceae, but we do more functional genomics and genetics. I'm wondering about the reticulation as it relates to traits that were used to distinguish obviously related species, and whether or not you can go even further and ask questions about reticulation is affecting interpretations of independent evolution of traits, like evolutionarily novel traits, for example, like inflated calyx. To what extent might this impact those interpretations to the point where maybe there's fewer or no independent evolution events for particular traits?
So that it would be like it might have evolved once and then been lost. Is that the—
Well, it may have evolved what looks like independent evolutionary events currently based on the data.
Yeah.
Would the discordance be masking fewer independent evolutionary events, or may there perhaps have only been one?
It might be easier for me to answer because I'm at the interface between the morphological work by Rebecca Hilgenhoff, but Edeline, you might have thoughts too. I just wanted to say that in my view, it's pretty sure that this will not make any of the traits that we previously thought are multiple times into a single, if that helps. So many of the fundamental characters, very kind of basic characters like inflated calyx, no matter which of these realities that Edeline highlighted will be the truth, if there is one single truth, it would not mean that they've evolved only once.
Well, what about if there were a suggestion of 10 times? Could it actually be just 5?
Yes, that will happen. But just to say that none of the multiple times will go into 1.
That makes sense to me. Thank you.
And that's, for example, tubers, interestingly, they are so in remote parts of the phylogeny that no matter which of the realities, there will never be a single evolution of tubers in Solanum. Yeah. But yes, absolutely, the number and rate will obviously change.
Thank you.
Yeah.
Thank you. We had a question in the chat.
Can I quickly go back to the previous question? So you were talking about probe sets. I would recommend you contact Gustavo Arias. He'll be talking a month from now, I think. So he's also in Munich, like I am, at the population genomics department, and he's put a lot of time in designing a probe set, but looking specifically at Solanum chilense. But they tested it on hundreds of plants now, and they have at least an idea how well it works within the species. And also branching out on what you say, so not just for taxonomy, but also linked to my own research. What is the resistance capacity? What are the resistance genes, etc. So they've really tried a lot, and especially for the resistance, there are a lot of bottlenecks there, of course, because these genes are extremely complex, and you might lose a lot of sequencing power if you focus on them. But you can contact him, or you can contact me again. We've done quite a lot with probes design in these contexts, and I'm happy to help and think along, like depending on what your questions are. So
cool, I'll drop you an email.
Yeah. And Gustavo is part of this Solanaceae seminar. He's gonna give a talk in 1 month. It's in the schedule. Okay, we had a question here in the chat from Federico Roda. He said that it's a great talk. And he asked about do you think that the reticulation is caused by natural phenomena like hybridization, integration, selection, etc., or is something related with the phylogenetic analysis, the method? He's asking that.
So I would say, so obviously you need to know more about the biology of the species to answer these questions. And I'm not a Solanum specialist, but from everything I've read, for example, from the Leptostomonum clade and the evidence that we have that it's the fastest diversifying clade in the Solanum, then that part of the tree, it's possibly, yes, due to, we also know there's a lot of hybridization, integration that's going on often amongst these species. It's difficult to know how much there is, because that's actually studies that require to look more at the population genomic level or to dig in deeper. So for that part of the tree, yes. But deeper in the tree, I'm sorry, that's some sun. Deeper in the tree, that's really tougher to get, because as we go back in evolutionary time, it becomes more difficult to reconstruct this type of phenomena. And as I mentioned, we have a case in Solanum where we have extremely short branches. And so it might be actually very, very difficult to test whether this discordance is due to incomplete lineage sorting, hybridization, etc. We might never get an answer. Doesn't mean we can't try, but it's going to be more difficult. And this is why I mentioned also that maybe getting full genomes eventually might also deliver a better answer about what's going on down there at the base of the tree.
So many of the, just to expand from the kind of natural history side, what my knowledge spans, so Solanum is such a big genus that I only know this much most of the time. But for example, Anomalostemon, clade that Edeline highlighted as one of these ones showing incongruence and also very fundamental placement of it at the base of clade 2. We know very little about. We don't know chromosome number. We don't have seeds. It's a monotypic lineage, endemic to a very narrow area. So that we don't know whether there's any polyploidy or hybrid origin of that species. And another Recmandra clade, none of the species we have chromosome counts. Again, we don't know whether they're polyploids or hybrid origin. So some of them are just simply not known in terms of natural history. But then there's also that, if the notes are so short, what will we ever do? Things pointed by Edeline.
Thank you, Edeline. Thank you, Tina. Federico, for the question. Someone else? I think we have time for a last question.
Can I ask a question, please?
Here is okay.
Here is, well, congratulations, Edeline. And I just want to mention that you find a very interesting relationship among Ituberosum and Petota, the potatoes, because in the current phylogenetic studies, Ituberosum appears at the base of tomatoes and potatoes. But here you show it that are closer to potatoes. And if you take a look of the plants, really the three species of the Ituberosum clade look like potatoes. So to me, it was very interesting to see that you show it in your results, these relationships. And to me, it looks like you have there in the potato clay a very interesting place to try to respond hypothesis, because Ituberosum is Rhizomatosus, the three plants. One is in the Juan Fernandez archipelago, very far away. Then you have potatoes, two species, and then you have adapted to high mountains and very cold environments and very extreme environments. And then you have tomatoes in a kind of intermediate environments adapted to the extreme aridity, like Solanum lycopersicoides and Solanum sitiens, that is a very, very strange species that can grow in Atacama Desert or in southern Peruvian desert. So I think there if you would like to explore hypotheses about agronomic trait, resistant to drought, or frost, or abiotic or biotic factors, I think you have a great opportunity. Mainly, you have the crops there. So any help I can provide to you, welcome and good luck. And very, very interesting talk.
So thanks, Edis, for your comment. I actually have two points I'd like to respond. So actually, I'm actually doing a sort of analysis with the occurrence data set for that clade in particular, so the Petota clade. So using the herbarium occurrence data and the ecophysiological factors to identify herbarium specimens or populations that occur in the most extreme abiotic conditions, as a way to perhaps help screen or identify populations that would be interesting to look at for plant breeding. Because they occur in the most extreme places in the niche. So we've already thought about that, and I think marrying if we could find a way to marry the genetic resource, or find a way to get at the genetic resources, it would be really fascinating. And about the placement of Ituberosum in the target capture phylogeny. So I don't want to burst your bubble, but Ituberosum is actually a sample that I included through a transcriptome that was published in the Single Reads archive. And it's one of the two samples which I had a really low recovery rate, so less than 100 loci. So I would take that placement as sister to Petota with some, like, a big red flag, a high degree of caution, warning. But I mean, if anyone has better data on like genomic or transcriptomic data about E. tuberosum, we could easily go get the baits for the angiosperm 353 data set and check with more data if that placement holds up or not. Yeah.
If Leonie Moyle is still online, can I ask your perspective? Sorry, I kind of jump on you in the audience, but in case you had any thoughts, because you really explored reticulation and kind of incongruence in data sets in a very deep level. What's your view on, if this is what you would expect, right? Question mark.
Hi, I've been hiding here, but yes, I am listening. Yeah, no, it's exactly what I would expect. In fact, I'm sort of secretly pleased, although a little bit sad for you, that indeed, even at the very base of those splits, you see a lot of discordance. But you will expect to see that whenever you have very rapid speciation events. Even the basal discordance is almost certainly influenced by incomplete lineage sorting all the way back then.
Right. So for you, the level of the depth, evolutionary depth of it, that doesn't matter. You would always expect
that's right.
It has been rapid and
yeah, that's right. So if you look at the base of, let's say, the mammals, there's a whole set of hard polytomies down there, a whole set of unresolved relationships. And that's also thought to be due to very rapid diversification way back in time. I mean, hundreds of millions of years, right? So the scale, the timing of that process doesn't matter so much. All it matters is that during that period you had rapid diversification, and that will never be resolved. You're stuck with that forever. So
in a way, your mammal example is great, because in mammals you would argue, okay, it's not hybridization, highly unlikely. Perhaps hybridization in the sense that yes, there's been like incomplete lineage sorting between species, but what I'm saying is that the level of genome flexibility is so much less that when in plants we start doubting, oh, has there been genome duplication? Has there been hybrid origin of species? We could argue that even in mammals we find this here, there, everywhere. Could you use that as a kind of logic saying, well, incomplete lineage sorting more likely? Not quite, but I don't.
Well, I think it's something that's emerging from tons of different data sets, right? So you can look at the mammals, you can look at the base of birds as well. Any system where they've had a really rapid burst of diversification, you see this unresolved phylogenetic signal that's coming from having lots and lots of gene trees that just refuse to agree with each other, no matter how far back you go in time. So yeah, I loved your data. I think it's really interesting and I really love the talk. But I do feel sad because it's going to make it really hard.
In a way, our job as a scientist isn't to try to make it simple. So in a way, if this is now going to, enable us to say, well, look, maybe us estimating 5 transitions may be equally likely to be 10, like Zach's point, the question of, what does it mean? What's the uncertainty interval? That's going to be a good thing. We can then say where we are uncertain and where actually, even with these alternate realities, we'd always get 5 transitions, then we know it's pretty certain it's 5. So I see that we've been talking about it, that in a way, knowing uncertainty is better situation than being blissfully unaware of it.
Yes, there's a bliss in being unaware. But yeah, I firmly believe that it's a real biological signal of rapid diversification. It's exactly what I would expect.
Thanks.
Good talk.
Okay, thank you. Thank you for all your questions. Thank you, Edeline, for
I have a couple of questions.
Oh, okay, the last one, please.
Oh, I'm sorry.
Okay, just go ahead. Go ahead, Carolina.
Okay, just one comment about the thing what you were discussing. It's possible that we can, I mean, maybe we should stop thinking in phylogeny like a dichotomy, always a dichotomy, and start thinking like everything is reticulate. Not everything, but maybe it's dominant, because it's like we try to put everything in 2 pieces, always bifurcating and binomial. So it's maybe we we can start thinking in reticulation as more often and the way to explain the diversity we see. But that wasn't my question. My question is two questions actually. Is when you use the plastome, you said you use only the exons data. Why only the exons? There is any reason to skip the spacers? Because in the supermetrics you have spacers.
Okay, so you want me to answer right away, or do you want to ask your second question?
Yeah, the other question is if you could see any relation between the phylogeny that you build with the plastomes and the geographic distribution of the clades. I mean, in the points where there is a strong incongruence with the other data set, if you see a reason to this new resolution.
Okay, so I'm going to answer your comments and then your two questions. And I agree with you that we often don't consider reticulation enough in our analyses, but that also has to do with a methodological issue in that a lot of the phylogenetic comparative analyses we have are based on bifurcating trees generally. And so I don't know of any like methods at the moment that allow for ancestral character reconstruction on a like network or things like that. So if anyone knows about that sort of method, or because I think there are some people who are trying to develop it, and it will come. But at the moment, they don't quite exist. So it's a bit hard to explore this further if you don't have the appropriate tools. For your question about the plastomes and the exons, so I went a bit through it quickly because it's a bit difficult to present 36 species trees. But basically, I not only looked at the exons, but I also analyzed the introns and the intergenic regions of the plastome data separately. And I also analyzed everything together. And so it generally results from the exon, intron, intergenic regions, and all the data together, they're generally concordant, except again for the key nodes I pointed out during my talk. So again, depending on whether you look at the introns or the intergenic regions, sometimes you get a sister relationship of the morloids with clade 1, sometimes it's with clade 2, but it keeps changing around. And bootstrap support is not a good indication because they also vary from analyses to analyses, and sometimes it's super strongly supported in one, and then it's completely conflicting with the other species trees. So yeah, further evidence of discordance in a sense. Finally, I have not been back to looking at the relationship between the phylogeny of the plastome and the biogeography. I guess, I mean, there is a biogeographic analysis of Solanum that's recently been published by a new phytologist, showing an old world origin, sorry, not old world, neotropical origin of Solanum with diversification towards the neotropics in Australia. Correct me, Tina, if I've gotten it wrong. So, but I'm not sure how these different topologies we found would affect that reconstruction. It's a bit difficult to say because it's so deep in the phylogeny. So those are the areas where you would expect a lot of uncertainty anyways. So difficult to say at this point.
Similar to the morphology, that none of these changes, independent of which one of the realities would be true, if there is a single one, they wouldn't make the geography any clearer or more messy. It's kind of messy already. There's some basic old world, new world disjunctions, and then there's more towards the tips. So that, for example, wouldn't change at all in terms of old world, new world chumps or to Australia. Maybe within South America, this could make a difference. So how much of it is like Andean groups would start clustering together? That, in my eyes, when I looked at the position of Regmundra, which is a very Andean group or Andean coastal. So yeah, maybe within South America, we might see different, but mainly relating to these niche shifts, biome shifts that Edeline is interested, sadly, in a way, because that may become untenable, but in a way pointing out the nodes where the uncertainty is, i.e. if you sample both sides, you might find very different pathways in terms of abiotic stresses. So that point will still remain, but it's hard to know directionality.
Okay, great talk, thank you.
All right, that is all for today. Thank you to everyone for connecting to this seminar. I hope you enjoyed the talk. Thank you to Edeline again for that wonderful talk. You guys, we expect to you next week on September 11 for the talk of Luke Wheeler that is going to tell us about an integrative approach to studying the evolution of floral pigmentation using as model the genus Geochroma and the petuniclade. That's all. See you next week.
Okay, see you next week. Thank you, everyone.
Bye.
Thank you, Andres and Edeline.
Thanks, Rocío.